Our next speaker, Gaute Einevoll, is a professor of physics in Norwegian University of Life Science and University of Oslo, where he works on brain physics. He's been a partner in the large-scale EU Human Brain Project since its inception in 2013. In his research, he uses methods from physics to model nerve cells, neural networks, brain tissue, and brain signals. He's also written popularizing books on science and produces a popular science podcast, Sense in Science. And his talk today is titled Towards Multipurpose Biophysics-based Mathematical Models of Cortical Circuits.
Okay, thanks a lot. Can you hear me?
Yes.
Very good. So first, thanks a lot for the invitation to come here or to participate. Of course, I would have liked to be in Helsinki, but that's how it is these days. Actually, I have a strong connection to Finland, actually, because my mother grew up in Finnmark. So when I checked my DNA and one of these 23andMe, they said that I'm 1/3 Finn. So all my closest DNA relatives are Finns, actually. So anyway, so next time I'd like to be there in person to check out the relatives. So cheers. Let's see if I... So the topic of today is really sort of towards multi-purpose biophysics-based mathematical models of cortical circuits, essentially trying to do the physics program also for a cortical circuit, in particular using the mouse primary visual cortex as sort of like the example to start with. But I like to go back to the motivation for, at least my motivation for why we're doing this, because I like to start with this image from a stamp of this Norway's most well-known mathematician, Nils Abel. He died in, well, only 26 years old, almost 200 years ago, from tuberculosis. And he was really upset about medical science. Why can't they fix this silly thing? I mean, how hard could it be to sort of kill this disease? Of course, now we know it's a bacteria and it's we have antibiotics. So in our part of the world, we don't die of tuberculosis anymore. But if you come back from the doctor with a serious ailment for the brain, like dementia or like, say, schizophrenia, which afflicts a lot of people, then it's not so much medical science can do. And this is also was actually the topic of this commentary written by Stephen Hyman, who used to be the director of the National Institute of Mental Health in US, probably one of the biggest funders of research on this psychiatry. And they also said that what has gone wrong scientifically since we are not, I mean, despite all these efforts and resources going into this, that we have not been able to deal with this kind of psychiatric diseases. And of course, then when you have all these resources put in, you have so meager outcome, you should maybe start thinking, are we doing the research the right way? And is present day neuroscience done the right way? And I think one thing that has been lacking really is that we don't really understand the brain, and it's often difficult to fix a thing if you don't understand it. I mean, sometimes you are lucky, you can find a drug that sort of like is miraculously work. Maybe like the history of some of these very useful brain drugs has been that it's been tried out for something else and they just realized that it had a beneficial effect on the brain. But if you go to the understanding, I mean, this influential book by David Marr from like 40 years ago, where he operated with these 3 levels of understanding of the brain. And using the visual cortex as an example, I'll do that here. I think that's sort of certainly part of cortex where we know the most. So you can try to understand this at the computational level, algorithmic level, and at the physical level. And ideally, you would like to have understanding at all these 3 different levels, and you would like them to be interconnected. So And if you look at, there are several theories for how cortex works at the computational level. There are all these ideas about what we call efficient coding, predictive coding, like pooling of feed-forward filters, the V1 salience theory and thousands brains theory. And there's all these cool books written about it. And some of these books are quite old, and it's not like the ideas go away, because it's very difficult to establish which ones, if any, is used in the brains. I'm quite sure that some of these, at the end of the day, when we understand sort of the brain much better, we'll sort of find some good insights or good guesses from some of these books. So this, how do we go about sort of like finding out what theory is right and what is wrong? And then at the algorithmic level, you also have some computational level theories have suggestions for sort of accompanying algorithmic theories, particularly this pooling of feed-forward filters. That sort of has been used to make some of these programs, which are like one called HMax, which is quite well known from Riesenhuber and Poggio, like 20 years ago, where they have these filters that makes it possible to sort of the network to recognize, in this case, different shapes of paper clips. However, there's no link between this computational or algorithmic, or at least not a clear link between these levels and the physical level. If you start like bottom up, looking at the neurons and trying to hook them up in a network, it's very difficult to make contact with these models. It's certainly not the case that you can sort of use what physical models to at the moment to distinguish between candidate algorithmic or computational models, to at least not in a very detailed way. So what is this experimental situation? Well, until now, systems neurons have largely focused on what we call neural representations or receptive fields. So we have the field that's measured receptive fields, starting with the work of Jubel and Wiesel, found this very nice orientation selective and direction selective cells in V1. And now there's sort of like all this, and typically, so the knowledge is often we have all these different cells which are named after the thing they respond most to often, or what they sort of make them special. So beyond the primary visual cortex, we also have like whatever color cells and Jennifer Aniston cells and whatever object cells and all kinds of cells. So this, of course, obviously has told us a lot about how the brain does things, but it doesn't really tell how the brain, what is the mechanism for making these receptive fields? What are the connections? What do the neurons do? And how are they connected in order to make these neural representations or receptive fields? And also in terms of interpreting the to be able to predict what the brain does in different situations. There was this in 2005. There was this mini symposium at the Society for Neuroscience in US, and it ended up with many leading visual neuroscientists, and they ended up afterwards writing this mini symposium paper in the journal Neuroscience. And the first line of the abstract says, we can claim that we know what the visual system does once we can predict neural responses to arbitrary stimuli, including those seen in nature. Well, I think it's, maybe you can sort of discuss this, whether you agree with this claim, but regardless, at the moment, even with all these neural representations and these different types of models, we cannot really predict, we cannot use, for example, a receptive field fitted or found from measuring response to spots or ratings to predict what happens when you show a natural movie to the animal. So what you need, what you would like, is really the goal here is really to get what I call here the multi-purpose model. A model that can be used for many things, not just sort of predict one particular aspect of one particular experiment. Of course, if an explanation from neuroscientists when they ask, why aren't you doing better? We're giving you all this money for many decades and still we're not well, we don't understand the brain and we have still big problems with treating patients. Well, we say that there's all these scales, which is hard, make it hard. That we have, and neuroscientists often work on one of these scales, spatial scales, at the level of molecules or cells or the whole brain. And it's difficult to connect these scales. And this is true. It is difficult to connect these scales, but there are also examples from physics. The one that we learn in quite early when we study physics is this: how you can describe a gas both in terms of thermodynamics at the sort of macroscopic level, like a box of gas, or at the microscopic level in terms of molecules, movements, and Newtonian mechanics. So here, in that case, like Boltzmann and Maxwell showed how to connect these scales. In the brain, it's a bit more complicated because, I mean, if you take one of these gases of, say, oxygen molecules and replace one of these oxygen molecules with a nitrogen molecule, then nothing happens, right? It's not like nothing happened at the macroscopic scale. So the scales are not extremely intimately connected. While you can change one molecule, like one nucleotide base in the DNA, and suddenly you get the different eye color or something. So there you can sort of really have a really tight connection. However, there are other fields in science where, or I would say science, technology, engineering, where people have been able to, or scientists have been able to bridge these scales, even though they are not connected in an intimate way. And my favorite example is this modern numerical weather prediction. Because there you actually have this connection between things happening at the 10-meter scale and up to the global scale. So it's many, many orders of both spatial scales and temporal scales. And this is the problem of mathematical numerical weather prediction is a bit similar to, well, that's like the standard physics approach. You put up a model, you have this idea about how I mean, the physics of weather, I mean, pressure, and like you put up differential equations for pressure and whatever, rain and wind and stuff. And then you need some data about what are all these parameters now, and then you can use the laws of physics to compute what happens in the future. And this was suggested as a program maybe in 1902 or something, but it took until the 1980s because before this was practical. Mainly because we had strong enough computers to be able to predict weather actually before it happened and not just after it happened. And interestingly, it's a steadily improved quality. I mean, you actually now predict 1 day better into the future per decade, meaning that the accuracy of prediction 6 days into the future today is like 5 days into the future 10 years ago. And this is this fantastic collaboration between people we haven't really heard about, even though we all get this weather forecast on our smartphones. So these are people with all these different, well, national meteorological institutes and this regional and international computer centers and so on. So I'm not quite sure. This is the weather prediction I found from Helsinki last. So it's supposedly, well, not too bad weather in Helsinki. I'm not quite sure if that prediction turned out to be right, but maybe some of you can look out the windows and see. Anyway, for the brain, if you look at the models at the physical level, if you want to do this program for the brain, we have this one point going from the molecular network to the neuron, where we are doing quite okay. Starting with the work of Hodgkin and Huxley, we know how to bridge between like ion channels embedded in the membrane and what they do, and they are sort of like essentially just a little bit of it's a protein tunnel, or like proteins together making a tunnel. So it's sort of like a link between what molecules are doing, proteins are doing, and the currents going into the neuron. And that is something we can put together to use to predict what the neuron is doing in terms of this salient dynamical variable, the membrane potential. So this is actually a fantastic thing. So it seems now that we don't really know all the details of what's going on inside the cell in terms of the molecular network in order to predict what like the signaling properties of neuron. Interestingly, though, there's been like this really cool work where they have been able to model how molecules and molecular networks make a cell work. This is this work from Marcus, what was this, Covert or something, at Stanford. So they made this model. I think it was like many thousand differential equations, which were sort of extracted from literature, and they were able to make this little bacterium divide and run through a normal life cycle and divide just from in the mathematical model. But that's not something we immediately need to, we just take for granted that this cell is able to do what the cell is supposed to do to stay alive. And then we look at what's special about what is the signaling properties. However, if you're going from the neuron to the neural circuit, it's much harder, or we're not doing so well. Well, we're doing okay in the sense that we know the basic laws, the rules for putting these neurons together. We have good models for synapses, and we know sort of like how we know in principle how to put them up in a network. But in practice, it's very difficult to make a network that actually mimics a particular like piece of visual cortex for a particular animal. So we have has some nice generic studies of what networks can do, like in how to get like regular firing patterns and so on. But the main problem is we don't really know, we know the principles, but we don't know all the parameters maybe. I mean, we need to wait to set all these parameters to get all this biological information needed to put up a sensible or a model, a network model. So that is sort of the challenge now, and we, I mean, I'm part of the Human Brain Project. So this is a very nice international project. We also collaborate a lot with a group in Finland, with a group of Maria-Lena Linne in Tampere. She's also doing excellent work within the Human Brain Project. But here we wrote a paper on the why we need to do brain simulations, because this Human Brain Project has been a bit misrepresented about what the goal is, and the goal is not to sort of put all neurons together and in a network and hope that this sort of automatically gives a sensible cortical behavior. I can assure you it doesn't. So the goal is for brain simulations is rather something is actually to make what I can say like a mathematical tool for testing hypotheses. And we made this analogy to an analogy I liked about how when Newton Newton's idea about the movement of planets. I mean, this was like the greatest year of physics, 1685 or something, whatever, when he published his Principia. And he had this idea, this physical idea, his hypothesis. Well, what about if these masses, if the planets, say, move because masses attract each other, so that the planets move around the sun because there's an attractive force between the sun and the planet? Well, that's formulated here. That is like inversely proportional to the square of the distance and proportional to the masses, and then some constant. And then he needed the second thing. Well, what happened to the Earth or any planet when a physical force is acting on it? Well, it can accelerate, F equals ma. So how did he test this hypothesis? If you just look at these equations, right? I mean, how can you, it's not so easy to make predictions about planetary orbits just by looking at this, right? So he needed math. So he needed a mathematical tool so that he could test this hypothesis. Well, and then, of course, then he invented calculus so that he was able to test his ideas. And actually, the first calculation was done geometrically, but then he made calculus to make this kind of calculation simpler, and this is how we learn to do it today. But the point is, without mathematics like calculus, he wouldn't be able to predict planetary orbits or the like ebb and flow of ocean water due to the moon and so on. So you need this mathematics to test whether the idea is right or not. And this is sort of what we do, like why we need brain simulations, because Newton could do this sort of like with a pen and paper, test this hypothesis. That's just impossible. If you have an idea, I think this this T-type calcium channel is particularly important for something. Well, I mean, how do you test that out? Well, in a model, you need to be able, well, you need to have a model that you can sort of make changes to this channel and then test it out. And for that, you need a big like infrastructure. You need big computers, and not only big computers, you need also like models, like good candidate models, and a lot of data then. So what we pursued sort of as an idea, or like as a our project, or what we suggested is that if you want to understand, say, the primary visual cortex of mouse, and the mouse is of course now a favorite test animal because it has all this it sort of is a mammal, so it's sort of like the piece of cortex of a mouse looks quite similar to a piece of cortex of a human, but makes it more interesting. And of course, with mouse, it's also possible to do all these genetic manipulations just to probe the system better. And the Allen Brain Institute have then also done an enormous amount of very important work to map out all the connections in the not only the primary visual cortex, but the whole visual cortex, not only connections, but the neurons and all kinds of measurements, which is sort of like it's an extremely fertile data set to use for modelers. But how do you sort of find out if you have these candidate models? For one thing, you need models at different levels of detail. So this is like biophysical detailed neural models, like neural networks in terms of that. But that's not the only thing you need. You also would like to have like a more core or like a simpler description, maybe with point neurons, because it's easier to analyze. It's also much faster to simulate, so it's much easier to do science with it. You can try out many more parameter sets at level 2 than at level 1. And then also you would like something at like maybe at the firing rate in terms of populations, because then they are actually possible then to, they come in different variations, but some of them are actually even possible for, like, if you're a little bit trained in mathematics and physics, to actually get some intuition about what the network does. So it's not only like if you do simulations at level 1, it's sort of like a black box and you can compare with experiments. But that if you have like a simpler model that is sort of some which has sort of like contains some of the salient properties, then it's actually together these form a nice combined, well, a nice understanding. But then we need, of course, also experimental data to compare and falsify the model. You need experimental data to put up candidate models, and you also need it to test predictions from the model. And then you can go back and forth between experimental data and models. And this is sort of like the standard physics or engineering approach. But one important thing is that traditionally, if you want to falsify models, we have only used spikes, measurements of action potentials, because they are quite easy to interpret in the sense that a spike is actually an action potential from a neuron which is close by the electrode. But we have all these other measures that is also containing actually a lot of information, like the local field potential, which is a low frequency part of the signal you measure with an electrode inside the brain, or ECOG and EEG and MEG and voltage sensitive dye imaging, and also calcium, for example, 2-photon calcium imaging. So we need to make this be able to compare. You also take advantage of that, these signals, when we sort of want to test or validate our model and try to identify like a candidate models which are better at explaining experiments than others. So what is the present models of primary visual cortex? Well, most of the models have been at, I would say, at level 3, often just considering 2 populations. Like typically there's layer 4 maybe, where they get input from the thalamus. And then they are typically like 1 or 2 populations, 1 or 2 dynamical variables. And they're typically what we call single purpose, not multi-purpose, and predicts like orientation selectivity, direction selectivity, contrast invariance, and things like that. But they often fail on, I mean, they're typically not sort of like able to predict more than one thing if you tune the parameters. And then at level 2, we have maybe this cat V1 column model, 80,000 neurons for like 1 square millimeter of cortex, maybe like particular cat cortex. And, and this was tuned to actually not to do any particular function. It was mainly, it was like lots of data taken from literature regarding the connection sizes and EPSPs and so on to set up the network. And then they tuned it so that you had like a sensible background activity, maybe this asynchronous irregular thing. But a very nice thing about that model is that it's publicly available. Everybody can download it. It's available several places and play with it. And that's what also people are doing. So it's very, very useful with network models, or in any kind of models actually that can be used by others. And now the model that our group is very interested in, and we're collaborating with friends at the Allen Institute, is this model, the new model that came out for the mouse V1, based on all this experimental data recorded at Allen Institute in the group of Anton Akipov, 230,000 neurons, and it's really bottom up. I'm coming back to that, how they made it. It's sort of like the state of the art, I would say, certainly in terms of like visual cortex models, in terms of taking this bottom up program seriously. And then there's also a variant of that model, which is in terms of leaky integrate and fire neuron models, neurons, which makes it much more practical to, this is like, I think it's a factor 5,000 or something faster. So this is something you can play around with much easily than this biophysically detailed neural model. But anyway, at the moment, we don't have a connection between, I would say, like these level 1, level 2 models and these firing rate population models. They are sort of living quite separate lives. So this is something that we need to bridge. Multipurpose models is then that models that simultaneously can predict experimental data for different stimuli and also maybe for individual trials and for different measurement types and for different brain states, ideally. And the key thing is that multipurpose models must likely be high dimensional, meaning that there are many dynamical variables, many differential equations, like in real brains. So it probably will have to be maybe at level 1 and level 2, certainly they are high dimensional. So they are candidates. And maybe you need that kind of model to make them multi-purpose. But they should be reducible to several single purpose models at level 3. So sort of like firing rate models. And in order to do this, we need big collaborations. The Human Brain Project is working on making it possible for neuroscientists everywhere, first in Europe and also in the world, to actually get access to all this data and data to make models, get access to other models and previously made models, and also get access to supercomputers so that you can sit like in a mountain in Norway. I like to sit there in my hut there, and do simulations if I want at some super duper computer somewhere down in Europe here. So this is fantastically, like fantastic possibility that lies in this, that you can now do like neuroscience all over computational neuroscience using all the data available. Just if you have access to internet. So the number of brains, human brains, like think about China, Indonesia, all this, India, in addition to Europe and US, all these countries that can now have these young, eager brains that can start working on these models. And this something called the e-brains infrastructure within the Human Brain Project will make this possible. That's the plan. And in 2023, when this Human Brain Project is scheduled to finish. Anyway, one thing that came out of this other large-scale project is the Mindscope project at the Allen Institute for Brain Science, where they have really provided an enormous amount of very useful experimental data, but also combine this and use them to make this neural model state-of-the-art neural model for the mouse primary visual cortex that I mentioned. Just like Roger Troub, he's a quite well-known computational neuroscientist. He said, I mean, he introduced this huge model, 3,500 neurons. It's sort of still used, even though it's like 2 orders of magnitude smaller than the new model from the Allen Institute. Said when he made this model for the somatosensory cortex, said that the greatest scientific challenge, perhaps in all of brain research, is how to understand the cooperative behavior of large numbers of neurons. Any model, even of a small bit of cortex, is subject to difficulties and hazards. Limited data, large numbers of parameters, the expense and slowness of the necessary computations, and serious uncertainties as to how a complex model can be compared with experiment and shown to be predictive. And the above difficulties and hazards are too real to be dismissed readily. And I certainly agree that this is too real to be dismissed readily. I think, however, there's like now is a good time to make like a real effort to try to do this for the mouse primary visual cortex. That's at least where I spend my time now, most of my time. And so we have started this collaborative research project where we, together with people at the Allen Institute, where we want to build this sort of like to take this Allen V1 model, which can be sort of considered like a skeleton model in the sense that not all the parameters are, it has sort of been tuned to account for some, like, or in direction selectivity, some features of spiking, but it doesn't sort of by it. I mean, this is like a starting point for further refinement. So now we're working on using most of the data from the Allen Institute to where we compute both, where we compare predictions from the model, both for the spikes and the local field potentials with experiments and try to do this iterative back and forth to get to a model that is sort of can explain more experiments. So what is the scheme for finding this? It's very like a very, in some sense, standard scheme. I mean, the people at Allen now has made this first version of the model I mean, and this published in this paper based on prior data. Now with that model, you would like to test it on different things. You have to choose like stimuli, input data. And an important thing is that if you have like measurements of we can record the inputs to V1 like we can in this case because they have electrodes in LGN also. Then you can actually test your model with data with like some recorded input data and also feedback data from higher visual areas. So the input doesn't necessarily have to be completely modeled. Anyway, so we have this model responses that model that depends both on the parameters and the input, and then we need to compare with these brain signals, and the Allen Institute just released sort of like this, I think, data from 60 mice with this fantastic with 6 neuropixel probings, 6 neuropixel probes, each of them has sort of like 380 contacts or something. So this is really state of the art, and that sort of penetrates like many of the main visual areas, as well as these LGN and other areas that sort of more feeds into the cortex. But then you have to make a decision. What do you want the model to predict? I mean, do you want to predict is spikes more important than LFPs, or LFPs more important than spikes? So what do you, and it's also a question, what do you want to predict? There's a big variability between mice when we look at the data, experimental data. So should you like to make a model that predicts the experiments for one mouse, or like some average mouse, or what should you do? Anyway, if you have decided on the target, then we have to compute brain signals, not only spikes, but LFP and other things. And then compute the deviation between your prediction and your target, and then check whether are you doing as good as is the deviation smaller than the criterion that you set. And if no, then you go back and forth in this back and forth fitting loop. And then if you eventually come to a point where you're happy, then you say yes, and you sort of declare victory, at least preliminary victory, and say, well, this is sort of a model that is sort of can explain these experiments to the accuracy that we sort of have. I want that this. So this is sort of like the best V1 model given the present data. It's not the end of the. It's like not the end of the like pursuit for better models. There's you can test it on new things, and also there will always be new data coming in. For example, there's new prior data now coming, new data coming from the Allen Institute on how synaptic connections, and also this fantastic set of electron microscopy where they sort of have this data set of all the, I mean, this at the really microcircuitry of the mouse V1, which tells you something about where the synapses are, where they're placed, and synapse numbers and things like that. So this is not like a model that like a game that you sort of this will go on for a long time, and hopefully then better and better models that is sort of more and more multi-purpose and better and better. One important thing is that something I've realized is that you how do you, I mean, the hardest part of this project is really to fit the parameters. It's not simple to, it's simple often to fit a statistical model or like just like a mathematical function to a curve. That's simple. Then you often don't need so many terms. But if you want to fit a mechanistic model so that the only thing you're allowed to fiddle with are like maybe synaptic weights, the things that you don't know, sort of synaptic weights and so on, and it's very hard. But one thing that helps us maybe is that we have this intuition that if you have like a fitness landscape, or this should be like cost, not fitness, then with few parameters, then you maybe start with with a parameter set that gives you like not so good fitness, and then the game is sort of trying to change the parameters to get to the bottom of the, like in this case, get this global minimum, and then you sort of say, well, that's the best model. Of course, the problem is if you start up there, then you can get stuck in this local minimum down here. So this is a good thing, is that it's a unique optimal model, but you get easily stuck in local minima during fitting. However, models with many parameters, like the V1 circuit, has typically, depending of course on the cost function you put, but typically with the cost function will have a very flat bottom. So if you start here, you will end up maybe here. And if you start there, you will end up in another model, but that is maybe performing equally well. And that is probably close to, I mean, when you see two different mice, I mean, if at the level of synaptic weights, they are between two neurons, they're not going to be identical. I think two mice, like the primary visual cortex, may function equally well in terms of what it's supposed to do, but it's not the same model, all the parameters are not the same. So there are many optimal models. And an important thing is that if you are like in, say you have a 1,000-dimensional model, a local minimum there that you get stuck in would mean that you have like, you're in a valley in all 1,000 directions. And what are the chances for that? I mean, there's like 909 uphills and one saddle going down, then it can go down there. So you never get stuck in the chance of getting stuck in these very high dimensional models are low. So this has been speculated to be why these deep networks are working so well. That like two identical, like in this case, convolutional neural networks trained on the same images, the similar performance, but trained parameters will be different. And actually, this is something that I discussed. This is the idea actually I learned from Terry Sanofsky, and I wrote a paper about this unreasonable effectiveness of deep learning in artificial intelligence. And I also covered it in this podcast, Sense and Science. So brain signals, that is, we need to be able to compute brain signals. And that is like a different, and the idea is to have all these kind of optical and electrical brain signals, both measurement inside the brain, invasive, and non-invasive like EEG and MEG. And typically these are just like analyzed or statistically. But an important thing to know is that we know if you have a mechanistic model like we have at this level 1 model, we can then compute all the contributions to the different signals, at least when it comes to particularly the electrical measures. but also the optical measures like the spike and LFP are just weighted sum of transmembrane currents in the soma region. Same thing with the MEG. And for like the optical, like voltage sensitive dye imaging, then it's actually what you measure is the weighted sum of the membrane potentials close to the cortical surface. But it means that if you have a biophysical model of this neuron and these neurons, we can compute the contributions. So we need to work out these mathematical connections between the neuron dynamics and the different experimental modalities, the like sort of like the measurement physics. And this is something which has not been, I think, has been underemphasized in computational neuroscience. That often the models have been like point neurons computing spikes have been compared maybe with measurements with EEG, and that has to be wrong because this like because of the just simply have neglected to take into account the measurement physics. The EEG signal is not proportional to the firing rate. I mean, it depends on other things. The point is we know how to do it, so we need to do that. If you have a candidate mathematical model for say network dynamics in the brain area, you need to be able to predict all these different measurements. And we need tools to do that, and we have made this LFPy tool, which is now in a new edition, so we can also do some simulation actually of also now EEG and MEG, and also MEG then, which of course is particular interest in Helsinki. And and this is sort of like, there are also good books written about this. Virenque wrote a chapter in this handbook of neural activity measurement. Now like two of your, Risto Ilmoniemi and Jukka Sarvas wrote this excellent book about brain signals. So this is sort of, we have to take this measurement physics seriously, but this is sort of like you have to do it right, but it's not conceptually difficult, like for example how to fit parameters in the neural network to make it behave sensibly. That's much a harder conceptual problem. So we have worked a lot on the LFP, the modeling of extracellular potentials. It's like a follow volume conductor theory. I won't spend too much time on that now, as I see. My time is running out. But we can then compute things like the extracellular potentials. In this case, it's an extracellular. The green thing is the extracellular potential computed around 5 neurons. And the green thing means that it's very low. And you see below there are some blinks and flashes sometimes. And that's just the spikes that you sort of pick up when there's an action potential in the neighboring neuron. And and we can compute all these all the different features. For example, LFP is if you have like a single neuron and that receives a single oscillatory current about the apical dendrite, you for example get this very nice dipolar pattern that that is sort of like what you pick up with EEG and MEG, that is essentially. And if you do it at 100 hertz, this just illustrates the importance of doing the measurement physics right. If you do it at 100 hertz, you don't get the same thing 100 times faster. You get a very different pattern, and that has to do with the cable properties of the neuron. And we can also do the same thing now with the computing, with the EEG and also MEG. This is sort of like if you sort of have like one active population in the human cortex with an electrode in the middle, you can measure the LFP and the spikes, and then you have like ECOG and EEG measurements. And this is just an example of, you have 10, if your population is 10,000 like biophysically detailed neural models, what we call the haystack, because it's based on the hay neural model, then we can see that here we compute both the local field potential measured at the core of this haystack, and this is then the EEG and the MEG that you would measure with a radial, like a radially oriented population, and this is with the tangential populations, like if you're in this sulci. Point is, this is not a very exciting network, but we can compute the signals, and what we see is that you only get a strong signal when you either get like all the input at the top or at the bottom. If you have it all over the place, like now, you don't get a strong signal at all. So you can hardly pick it up with you wouldn't see it as an LFP or an EEG or an MEG. So the point is that even if it's evenly spread out like this, you could have the same spiking activity, but these population measures will be completely different, and not only different in magnitude, also have different polarities. So this is something we just have to do right in order to compare with experiments. Also, you can also do this from point neurons. And that's not, so if you have point neuron networks, you can also compute these signals, even though it's not, doesn't follow automatic in the sense that point neurons by themselves do not have extracellular potentials. They don't have like this, don't generate current dipoles. You need at least have two compartments to make a neuron model with a current dipole moment. So, but what you can do is what's this trick, we call it the hybrid trick, where we compute first, this is the potentialismal model, compute this, the network dynamics and the spikes with nest, and then we store all the spikes, in this case for 80,000 neurons, and then we replace all of these 80,000 point neurons with 80,000 biophysically detailed neurons, and then we can actually replay it, and then we can compute things like the LFP measured at the central core, and also the EEG at the top of the head, and MEG also if you want that. Here it's not, there's like an input from thalamus at 900, and there we can see, bing, it gets this strong LFP and EEG signal. So the point is that we have a forward model which starts with neurons. I should have mentioned that we just have a paper coming out, was just accepted in NeuroImage, where we show how to do this to do this feed-forward modeling of EEG and ECOG signals based from neurons and networks. So yeah, I'll just briefly say that we're now working on this Allen model to try to tune this model to both predict LFP spikes and LFPs. And this is a very detailed model, 16 different or 17 different model neuron types involved, 4 or 5 layers, and in 4 of the layers we have 3 different types of interneurons, and they are all then extracted from found from experiments and recorded at, all this data is coming from the Allen Institute then. And so then we work on this, trying to tune this model against spikes and LFP. And here's just an early example result where this is just like an LFP from an experiment, and the CSD, that's the current source density. This is the flashing spot, which is turned on at 50 milliseconds. And this is now, this was just an example when you're working on when you compare experiments, or like this is the experiment, this is the particular version of the model, and we see that some features are reproduced, but others are certainly not. And this is just like some early example results where we both then compute the LFP and then this current source density and also the firing rates. And it's not so easy to make contact, or to fit experiments. And then you have the issues, what experiment should you fit when there's so much variability between animals. I want to just say a very quick word about another project we were working on. The big revolution, or one big revolution in biology, has been this, that we can map out the human genome. And so now, 23 chromosome pairs throughout 3 billion base pairs. And the second, and maybe the most amazing thing, is it's now so cheap to do this DNA test. Like I could find my Finnish connections by just sending out little spit sample to some company in US. And they say now, if you want to do this thing if you go to hospital to get this done, it costs more to park your car at the hospital than to do the test. So that means that you can test this like standard on many large groups of people. And my colleague Ole Andreasen, he has worked on with psychiatrists, so he is involved in this consortium where they have together. So the limiting factor is sometimes the number of patients. So I have looked into have this consortium where they had I think the first study that came out in 2014. It's like 80,000 test subjects, for which about half had schizophrenia. So then we could look for, okay, what is there any particular aspect of the genome, one of these SNPs that tells you something about the chance of getting schizophrenia or not? Because it is an inheritable disease, meaning there's like both inheritability and genetic variation. And what they found in this first paper from, I think, 2014 is that they could identify these positions on the genome, but it's not one or two, which would have been nice, because then you could have maybe done something with that, like it would have been easier to deal with in terms of genetic therapy and so on. But if it's like 108 positions, how do you find out? I mean, how do you test that things? How do you start fiddling with 108? It's like 108 possible like genetic variations, and it's like that's like a combinatorial explosion. So that's not possible. However, if you look at this, what do these positions along these positions that was found here, what do the DNA around there encode for? And then you found that many of them actually encode for neuronal excitability, meaning how easy it is to get the neuron to fire, and also synaptic function. So then we have sort of started to investigate this in a model where there it's much easier to change like many variants at the same time. And this is, I think this is a work that's been pioneered by Tuomo Mäki-Märtynen, who has been also came from the group of Maria-Lena Linne, and has been working with us in Oslo for some years on actually doing this kind of looking at effects of like the genetic variants on what you could sort of like on the things that you can like on electrophysiological properties, because there are some findings like in the EEG signals of schizophrenic patients, and in certain situations you can see a difference between like schizophrenic patients and controls. So there's actually possible then to make this circuit model and then do genetic variations sort of corresponding to what you could expect maybe then with the schizophrenic person, and then compute and then compute EEGs and then see what we see and compare with like the experimental findings. And actually this paper, last paper that came out in last cerebral cortex last year, we actually sort of were able to sort of see some certain increase in power in delta oscillations measured in EEGs and compare that with models. So this is sort of like a new way to actually get the genetic information into our like computational neuroscience scheme. And we have sort of like this is like this like what's it called, this suggested this thing called biophysical psychiatry. And it was like a paper last year where we sort of presented this idea of sort of trying to use this sort of like this kind of modeling approach to highlight maybe like what could be the causes for complex underlying these mental disorders. So the last thing I'll just show is this, or if you look at this physical level multiscale game that we're trying to play, now we also have a way to get genes into this whole activity, whole project. Because if you look at humans, I mean for animals and mice, we can have access to experiments at all these different levels. But with human, and you can sort of measure the genes, and you can also, what you also can do, you can study these neuronal cultures like this stem cell, using stem cell techniques. So that's an approach to learn something about the neurons and neural circuits, but still that's outside the body, so it's a little bit difficult. And then you have these non-invasive techniques, right? So modeling would have to be that's the hope, at least, that model can help to bridge these sort of scales also eventually in humans. So where we have all this targeted data from animals that can help sort of to falsify this or test this or develop this scheme. So thank you. That's what I plan to say. I went a little bit over time, but I guess it's we can just have a little bit less time for discussion. That's how it is.
Thanks for the very exciting talk. Yes, there's a little time or no time for questions, but we can take one. There are plenty of questions about the details of your work as well, but there's one very much upvoted question, which is more general. So I'll ask that instead. So your collaboration with Allen Institute is a fantastic example of open science. And would you agree that the issue of competition between research groups has been a major obstacle to open research? And would you have any advice on how to?
Yeah, no, it is really, it is sort of a little bit strange, this thing that people have, I think especially this culture of not sharing data, I think particularly among the experimentalists, is because it's, it's not been healthy, and it's a little bit strange that if you look at the Allen Institute, which is now then really pushing this open source, and there they make data available before they have sort of almost looked at it themselves, the scientists there. And this has been decided by Paul Allen, who funded it until he unfortunately died a couple of years ago. So it was a little bit ironic and paradoxical that the only data that was openly accessible almost was funded by a private person, but the ones that was funded by the governments and the taxpayers, that was not accessible. So, but I hope that it's because in some sense when you do a modeling, I mean, I would like other people to use my model. So I mean, the idea of not, I guess I could keep a model and say, well, maybe I can find something really clever with it that I haven't thought about. So, but so that I shouldn't share it. But I think sharing is in some sense, it should be the natural thing, because if we are sort of, even in terms of like, if you want to have it's all this work that we put into our science that we don't want to share it, and that's why you want this to have an impact, both for like personal reasons that you want to, like for personal vanity reasons, but also just to try to improve science. So I think, but I hope that is a cultural thing, and I think many good things comes to those who share. I think it's I think people have tried it in practice. They get much more positive. And it's also another thing. It's also, I think, the animal rights aspect. I think it's you should really make sure we need to do an animal experiments, obviously, but I mean we also owe it to the animals to get as much new knowledge from each data set as possible. So sort of just doing all these experiments and not just looking for one thing and publishing something and then just let the data die on a hard disk. That's not acceptable. And I think, of course, I mean, in terms of, we always say that in the grant applications that, well, this is important for this and that disease and so on. And this if you told the patients, well, actually, I'm not going to share my data to promote science because for like selfish reasons, it's not really acceptable. So it's just a culture that has to change.
Thanks. We'll have a short break and we'll continue in 2 minutes with the panel discussion. So join us back then.
Thank you.